An honor to have you here, Professor George Church.
Thank you.
Would you like to start by having any words or slides or anything, or should we just dive into my questions?
Um, I can pull up slides if in response to questions, maybe that's a possibility. We can just dive right in. Yeah.
All right, all right. I would love to hear about your story. How did you get into this? How did you end up to today, being a leader in genetics? And
Uh, well, I don't know about that, but I got here from Florida, where I was not really getting educated in science at all, in school, but as a result, I had to kind of learn it on my own. I didn't have any role models, and then I moved up north for high school, and suddenly it was much better, and I got really entranced by so many different fields I didn't particularly want to specialize. I did for brief periods of time, like for example, in my graduate thesis at Harvard, but for the most part, I was all over the place and still am. And when I became did a postdoc with Gail Martin in UCSF on stem cells, still working on those, and then became a professor at Harvard in '86, just 2 years after my PhD, and um, got interested in aging mainly thanks to Pedro de Magalas, who was a postdoc in my lab, not one of my first ones, but in the '90s, and been working on that ever since. And along the way, a lot of technologies we developed, next gen sequencing, next generation editing of genomes, and synthesis of DNA, and lots of developmental biology and aging recently.
tell us what we can dream of with the genetics. You're infamous for trying to bring back the woolly mammoth. Any projects like that you can tell us about?
Well, there's a few projects that are heavily based on engineering germline. Um, Human germline editing is off the table for now, but we can get it into humans via um, you know, developmental processes in a matched organism, in this case, a humanized pig. And so that's one big project that in a way also de-risks the conservation, endangered species and extinct genes that we're doing at Colossal, for a whole variety of different species now. And that's going very well. But the kind of the precursor to that was engineering a pig that had 69 changes in its genome to make it more human compatible, including knocking out all of its endogenous viruses. So I think it's certainly the first pig, probably the first animal to have all its endogenous viruses knocked out, and they don't come back. What we left protects them from superinfection. So that's a big deal. And then and now those are making into clinical trials for heart, liver, and kidney. Kidney is a straight up kidney transplant, which we've done one of so far. We've done one liver, which is the liver and heart are bridging models where you keep the patient alive long enough that you can get an organ, and maybe 80% of the ones that would die are saved by this bridging method. So yeah, so those are the three main organs that we have at eGenesis. And then we have in the pipeline methods of making it so that we don't need immunosuppressants anymore. That's something that is necessary for all human-to-human transplants, but we think we can make enhanced organs, both from an immunity standpoint, from an aging reduction standpoint, and reduced infectious diseases, like the knockout of all those endogenous retroviruses. So that's a sampling of projects that involve animal germline engineering.
Any chance we can bring back some dinosaurs?
So the record, it depends on how you want to do it. So the record for bringing back DNA, and we can get very high quality DNA sequence now from ancient DNA, all the way, the oldest DNA is 2 million years. But you can go way back. You can go back 3 billion years in terms of phylogenetic trees. And so you can reconstruct ancestral sequences that way. And that's been done. And then intermediate, there's a lot of developmental biology you can infer by the modern relatives of ancient. So I don't think there's any, depending on your goals, I don't think there's really any limit to what we can do. And most of these are mixtures, frankly, of ancient DNA, modern DNA, and synthetic DNA, the DNA that's better than both the ancient and the modern.
Fascinating. First, of course, we want to live longer, healthier lives so that we can see this amazing future. So you mentioned Pedro, how Pedro de Magalhães got you into aging. Like, but why? Why did you get into it? How were you thinking of it? How has your thinking evolved about aging until today?
Well, I think like most people, I was always interested in it as a young kid. I had great respect for my grandparents, and I just sorry to see them go. They but I didn't really think there was much I could do about it when I started. I thought that at a minimum, there were certain molecular biological tools we needed before we could do anything with stem cells, which was my postdoc, which my postdoc was a little ahead of itself. We needed to develop tools, and then we went back to stem cells. And stem cells, I think, have turned out to be a pretty key component of aging research too. It was still fairly primitive when Pedro was in my lab, but now there's been this exponential curve on all these molecular tools, as well as on making stem cells and differentiating them into basically almost any kind of tissue, including complex tissues and embryos and so forth. But still, the sine qua non, the ultimate for making really complex organs, is still requires normal development as we do in the pig. We can't do it in vitro yet.
Thank you. What might be underrated and also what might be overrated in the field of aging research and drug development?
Yeah, so I think historically, there was wishful thinking, which was, I think, reasonable. If you don't have the right tools, you got to do what you can do. I think there was a lot of wishful thinking and overrating of, nutrition, exercise, or eating or not eating something, drinking or not drinking, exercising or not exercising, all these things and even like simple drugs, where one small molecule, even if it's synthetic, it's still almost as wishful thinking as it could be something natural. So I think what we're realizing is that aging it's analogous to planned obsolescence. Obviously, it's evolved, not planned, but it's like planned where you have multiple organ failure, just like you might have multiple pieces of equipment failing in your car as it reaches its warranty. And it probably means that you're going to need multiple very powerful therapeutics. And my favorite powerful therapeutic is gene therapy, because you can really direct it very well. You can go directly from most scientific biotechnology know-how comes in via protein and RNA knowledge. And so you can immediately go from that to a gene therapy. In fact, we just in one series of weeks, we did 48 different gene therapies based on age-related diseases, thanks to Noah Davidson. So I think gene therapy is underrated. So was the overrated, underrated. It's underrated, I think, for two reasons. One is the perception that it's expensive. When we get to ethics of egalitarian distribution of technologies, but it isn't. It's only expensive. It's $4 million a dose for rare diseases, which can be much more cost effectively dealt with by genetic counseling, such as at Doria Sharim. But for things that affect everybody, that affect 8+ billion of us, then the cost gets down from 4 million down to around somewhere between $3 and $100. So like the COVID-19 vaccines, not everybody appreciates how they were formulated exactly as gene therapies, either as viral or non-viral gene therapies, and they were down around $30 a dose without government subsidy.
Yeah, I thought the AstraZeneca vaccine was actually even closer to $1 in some,
Yeah.
at least in jurisdictions, but maybe it was a small dose. So if you were to do a powerful gene therapy that would transfect into many tissues, it might be like $30, $50.
Right, exactly. So all of those vaccines, even though they're gene therapies, they were delivered intramuscularly and then let the immune system do a lot of the heavy lifting. So you're right. If you really have to get broad tissue distribution, it's going to be more, but not that much more. It's the
Even if it's
an economy of scale is what we're counting on, and aging is just like a pandemic in scale.
Yeah, and even if it would be hundreds of dollars or thousands, like actually the cost of treating the diseases of aging is so much higher, right? That any government would save money, and also, of course, by keeping people healthy and contributing to the economy.
Exactly.
There's also that benefit. So yeah, it's a no-brainer. Nobody would have
Talking
to
about trillions of dollars here per year. Yeah.
Maybe also a few other buzzwords, underrated, overrated. What about AI?
Yeah, so I think AI, I think is part of it's underrated, part of it's over. So the over part, I think, is things having to do with images and text, because I think that those are great if you're looking for, art and hallucinations and, inspiration or something. But if you want, a factual or innovative and factual breakthrough, hallucinations are usually not what you're looking for. So that's the overrated part of AI. The slightly underrated, at least in the world at large, not necessarily in biotech, is we've had a revolution since around 2019. We published a paper called on UniRep, which was one of the first protein design measures. And I think protein design has really taken off now, especially when coupled with multiplex libraries. So we call this MLML, for machine learning protein design, with these multiplex libraries, which can typically, we can use them up to maybe a million, maybe even a billion per animal. So you can use really expensive animals that normally there's no way you could afford a million clinical, preclinical animals. But if you can mix them together and put them into one animal, then you can get something that's far more precise than something you can do in silico. So that gives you a flavor of what's over and under.
Maybe another slight thing on AI, drug discovery. So could it help discover drugs, and also maybe predict results of preclinical and clinical trials, or help us?
Definitely. I think we're already doing a little of that. I think it's a little bit harder than the protein design, but totally feasible. Yeah, and I also didn't mean to say that writing text and doing images that are accurate and innovative are forever overrated, just right at the moment. Yeah, but I think small molecule drug design and utilization is going to be impactful very soon.
Yeah. you mentioned regarding small molecules being overrated. I agree generally. What about using them differently, not interacting with the metabolism, but rather things like bioelectricity? Is bioelectricity overrated, underrated?
Well, I didn't say small molecules are overrated. I just said that they're on their way. They're not quite as easy as protein design. Did you say bioelectricity? Was that what you were talking?
Yeah, no, I mean just small molecules regarding their impact in aging. I think
Oh,
uh, yeah, we kind of agree that they're quite overrated. Like people kind of were hoping that some metformin, some rapamycin
oh,
magic
that.
pill is
Yeah,
gonna, right? So
right.
those I think are a bit overrated in the field. Yeah, I just heard about bioelectricity from Michael Levin at Tufts University. Really fascinating stuff. So I was wondering if you have any thoughts on that. Um,
Yeah. So I think that um, the certain small molecules do work on bioelectricity defined in the conventional way, which is that they act on ion channels and with nerves, nerve muscle interfaces, and that sort of thing. I think bioelectricity is sometimes used in a more mysterious, like a mysterious force way, in the same, sometimes applied to epigenetics as well. I think they're not that mysterious, and anyway, I think it's a little early to weigh in on new forms of bioelectricity and epigenetics, but I think the conventional forms of them are already powerful enough, and we've got a lot of progress.
yes. Cool. What about organ replacement, maybe whole body replacement?
Right.
Uh, that side, overrated, underrated?
I think, well, certainly organ replacement, there is a big medical effort there, but there aren't enough organ donors. Um, and organ donation can put your own life at risk. It's one of the reasons. There should be a win-win where you can get an infinite number of organs or an adequate number of organs without people having to go through elective surgery. And I think that's happening. That's exactly what's happening, both with uh, human stem cell derived simple tissues. So far, we can even make brain transplants. So we have one going into clinical trials now of oligodendrocytes plus neurons that can restore demyelination like you get in multiple sclerosis. So and then there's Parkinson's treatment. So that there's a bit you can do with brain. And the reason I bring up brain is even if you replace every part of your body, you've got to keep at least certain parts of the brain intact because that's where your personality and your memories and the things that make you you. But I think even those core personal components can probably be replaced. I mean, it's not ruled out anyway, because your brain is constantly refreshing its memories and building, new memories. Even though there's not a lot of stem cells in the brain, there are a few in the hippocampus and other places. Um, you can introduce stem cells, and they can form they can become part of a network and learn the old memories. So I think that's a ways off, but I think it's still a possibility. So yeah, I think these pigs are capable of donating almost every part of the body. Right now, hands and faces aren't their forte, but almost everything else. So I think that we're moving in that direction, but it might be more cost effective to do it via gene and cell therapy rather than organ and body replacement. But they're all feasible.
Great. All right. So it still seems to me that this field is not progressing as fast as we'd like it. Um, Society sees something like a new virus come into the world like COVID, and it's, oh my God, it's an emergency, it's a crisis. Let's stop everything. Let's do Operation Warp Speed. We have something like a war, right? And then people are like, okay, let's make a nuclear bomb, Manhattan Project, make a new city like Los Alamos to quickly do that fast and end the war. How can our field get to just a complete, like a whole different level, something like Apollo Project, Manhattan Project?
Yeah, I think that part of the reason that we didn't do it is a difference in perception of how close it was. So towards the middle of World War II, it was quite clear how to make an atomic bomb. I mean, even though it wasn't guaranteed, in fact, it wasn't even guaranteed that it wouldn't start a chain reaction in the nitrogen atoms in the atmosphere, but it was close enough that it was simple engineering, just like going to the moon was considered engineering rather than science. Um, I think that wasn't the case for aging until recently. I think now it is, and we, in a certain sense, we have, um, whether we call it a Manhattan Project or not, we have exponential growth in technologies distributed all over the world. There are advantages of concentration, and I know that's one of the themes of Zelar.city and all these cities that we're considering here at this meeting. And there's no doubt that the high concentration of biotechnology, say in Boston, where you have almost every major pharmaceutical company has a research or administrative component there, and you've got Harvard and MIT and other great universities. That high concentration where everything's in walking distance is incredibly important. We'd like to duplicate that all over the world. So I think that would also help. But we've got the momentum now that both molecular technologies and aging and developmental biology in specifically are ready to take off. And I think it will be once you see that you can do it, then you can say, okay, now it's an emergency, just like COVID-19, maybe even bigger than COVID-19, because even though COVID-19 put all 8 billion people at risk, only millions died, even fewer than 1918 flu. But even 1918 flu is nothing compared to the number that are going to die due to age-related diseases, which is 90+% of us. So I think that crisis will be recognized once there's something we can do about it, which is about now.
Okay, I hope that's gonna happen. Our society is gonna wake up like, oh, now we can do something about it, and so on. I cannot help but worry, though, looking at the feedback loop. It takes decades to develop drugs, to bring drugs to market. Regulations seem to be slowing things down a lot. What's your take on these special jurisdictions, people going together, creating their own laws and regulations as a community, building a new city, yeah, or even just a special jurisdiction where people can do gene therapies much faster within a few months and bring drugs to market faster?
I think that's an interesting prospect, but it has to be treated with caution. I mean, even though there's hundreds of different places in the world that one can develop drugs, most of the countries tend to have fairly similar rules, and they're largely similar to the US FDA. Now, another powerful alternative to going offshore is to just move faster onshore. So, for example, the COVID-19 was approved in 1 year. Now, admittedly, a lot of R&D went into preparing for that moment, but you can't say that they had actually been working on it for more than a year, because it wasn't recognized. It just didn't exist. There was no sequence of the COVID-19. There was no emergency. So I think that was about a year, and I think that and that was new technology too. That people were not routinely doing messenger RNA vaccines or other gene therapies with intramuscular injections. So I think that we can reproduce that if we can make the argument that it's more of the same. I mean, the COVID vaccines were extremely safe and effective, and 6 billion people tested it, which is a gigantic clinical trial. So I think that we're going to see more and more gene therapies getting approved faster. I think we should aim for 1 year, but we need to be cautious that there are system level things when you're dealing with a system as complicated as developmental biology and aging, where two things might independently help, but together they might interfere with one another, or they could even together produce some synergistic positive or negative effect. So we have to do a lot of systems biology. We need to have better testing mechanisms. So I mentioned that we can now test millions to billions of different drugs or drug deliveries in one primate, which greatly reduce cost and speed of trials. But I think we are also getting better at making human organoids, and eventually we'll have, human embryo-like structures, and not through conception, and more complicated organs to test, and that will reduce the failure rate that happens in phase 2 and 3, where despite your efforts, there's either toxicity or unintended lack of efficacy.
Yeah, I definitely hope for a Operation Warp Speed for aging therapies. Although we're here just in case that doesn't happen, or even just to advocate for that, but just in case it doesn't happen, create a special jurisdiction where we can make a different regulatory regime and try if that works out better. Of course, safety number one. What other bottlenecks do you see in this field?
Well, yeah, in addition to regulation and concentration, which you described, and new models for testing, including multiplex testing in primates and multiplex testing in human organoids, all of those will be revolutionary impact. I think another one is just getting more people that are volunteering. So I'm kind of embedded in a culture where we're all guinea pigs. We sign up for clinical trials. We sign up for sharing our information. And the Personal Genome Project was a project that I established almost exactly 50 years ago. It'll be April. It'll be 50 years. Sorry, 20 years. That is a unique project in that everybody is sharing their full genome sequence, their medical records, and a whole variety of cutting edge multi-omics, transcriptomics, microbiomics, immunomics, so forth. And I think that if everybody on the planet could be a volunteer, some of them could actually make some money at it. Others will do it just out of the goodness of their heart. That would be a completely different culture in terms of science literacy, and it also would change the statistical significance of every study.
Feel free to take I can send you my personal genome as well, my whole genome sequence. Happy to. When would you take novel advanced therapies, gene therapies or whatever? Would you take them, and at what point? What would need to happen?
So, yeah, I would. I have been a guinea pig most of my life. My parents did experiments on me. I'm in the personal genome. I'm a member of the Personal Genome Project cohort of research subjects, as well as a scientist as well for that same project. I would be willing to take gene therapies as soon as we've made it through, these primate trials and human organoid models. We've got models now for Alzheimer's, bipolar, schizophrenia, autism spectrum, and even late onset things like late onset Alzheimer's. We can simulate in kind of an accelerated mode that's quite significantly different between Alzheimer patients and age-matched controls. So anyway, once it's been through primate and human organoid testing, then I wouldn't do it on a one-off where I just take the drug. It would be something where I'd be part of a cohort where we do blinded testing, and sometimes a nice model I like is crossover, where you'll either at the beginning or the end of the trial, you'll be getting the gene therapy, and then you'll swap. So people who got it at the beginning will get it at the end, will not get it at the end, and vice versa. And you won't know when you're getting the drug. I think that's important to avoid placebo effects and so forth. So anyway, under those circumstances, I would definitely be happy to be among the first to test a gene therapy.
Great. Last question for me. I have a lot, but we have a full room here, people even standing. So I'll invite people to start lining up here. So last question for me, what issues do you see in science? How would you like science to work if it was redesigned? I feel like we have an opportunity now to design a new system, especially with decentralized science. Think about like funding science, executing science, but also publishing science without permission, like just redesign a new system.
Well, what you just described is a redesign, and I've been supporting it for a long time. I was, my requirement for being on an editorial board about the time that I joined molecular systems biology was that it had to be open access, and that was the first of the Nature journals that at the time that was open access, and now I think they all have that kind of policy. So that's one part, but the other part is a lot of the money. So you can decentralize and open access as much as you want with government grants and with private funding and even self-funding, but a lot of the money still is going to be in the big pharmaceuticals, and it'd be nice to have and I think we're moving in the direction of having them announce their clinical trials in advance and then describe the outcome, whether it's positive or not. There was once upon a time, I think it's mostly gone, they would withhold the negative results, either because it didn't make them look great, they'd bet on the wrong horse, but more importantly, it would save their competition hundreds of millions of dollars, and they didn't want to do that. Say, don't bark up this tree, we've already done it. I think we need to share at that level, and so I think that's a less obvious part of decentralized science and sharing. You can't share everything, probably not with our current mostly capitalistic society and even nationalistic society, but everything we can move in that direction, I think, is good. And certainly when you can get economies of scale, which you do get for pandemics and for age-related diseases, that's something where I think a very important bit of science is getting the cost down for everybody. And I think that comes from economies of scale and novel technology. So, we brought the cost down of reading and writing DNA 20 million fold. And that was not just economy of scale, that was mostly innovation, completely changing how we did those. So I think these are important parts of changing science itself.
Thank you. Take it away.
All right. Thank you for a great talk. My name is Linus Peterson, based in Sweden. I'm a longevity advocate. So two super quick questions. First one, I would like to invite you to speculate on when the first intervention for aging will come to market. And secondly, what's the most exciting thing you are working on right now related to aging? Thank you.
Yeah, so the first intervention for aging, it depends on your definition. So some people feel that they have to convince the FDA that aging is a disease and therefore develop a drug specifically for that. Other people like myself feel like the FDA is already okay with curing age-related diseases. And if you have something that reverses aging, it should reverse diseases of aging one at a time or all at once. Doesn't matter. All the FDA needs is one, and it'll approve your drug, and then you can apply it to lots of diseases. What are we doing? It was the second part of your question. I've already, I think, expressed most of my biases towards gene therapy. In part, gene, protein, and cellular therapy kind of lump them together in that they can very specifically address protein, isozyme, isotypes, and alternative splice forms and so forth, that maybe have exactly the same active site. And so it's a little hard to do active site directed small molecules, but we have a bunch of the things going into clinical trials now, especially at Rejuvenate Bio. They have two triple gene therapies. One is the OSK of Yamanaka, looking promising, and the other is 3 bloodborne factors that are expressed at high levels from the liver but go everywhere. So I think those are your 2 questions. Yeah.
Awesome stuff. All right, next question here. Thank you so much, George.
You.
Hello, George. William James here from University of Maryland. What do you see the role of whole genome sequencing playing in aging, and what do you think it will take to get everybody sequenced?
One of my favorite questions. Thank you. Good question. So I think one of the things that we, kind of 2 parts of this, one of the things that we can do to help with aging related diseases is generic. It applies to everybody, and hence you can drive the cost way down. And so even though, since I've been an advocate of sequencing since the 70s, that seems to be naturally related to personalized and precision medicine, but the cost structure is much more impressive for generic medicine. And I think a lot of the aging related things that we've been talking about here could be applicable to everybody on the planet, maybe with small adjustments for personalization. Nevertheless, the genome is important because the generic is not going to work if you have something seriously wrong with you. And a lot of those can be anticipated through genetic counseling if you have whole genome sequencing of, let's say, in the matchmaking process, or in IVF clinics. And so then there are IVF clinics like Orchid Health that do whole genome sequencing on every embryo that goes through there, and that reduces spontaneous abortion, among other things. And then there's lots of serious diseases that you can prevent without gene therapy, without the expensive gene therapy of rare diseases, just by genetic counseling and some cases changes in diet. It just happens in newborn screening. So I think those are the 2 components: is the rare and the common have different strategies.
And what will it take to get everybody sequenced?
Oh yes, to get everybody sequenced is it's all, the cost is already right. It's already at the point where whoever's providing the healthcare, whether it's insurance companies or healthcare providers or national health services, whoever is doing that could get a 10-fold return on investment on average, because the cost of these rare serious diseases, and they're not that rare. Collectively, they're about 3% of deaths. Very expensive, in the millions of dollars per lifetime, and those lifetimes are extending, which means they're even more expensive, but in a very good way. But if that sequencing were done for as part of matchmaking, as it's done in the Doria Sharim community, it works like a charm since the 1980s. And so it's just a matter of educating, or a couple of insurance companies and national health systems taking that jump. But the cost is already, in the $100 to $200 range, and there's almost not even a need for genetic counseling, in the sense that false positives at the matchmaking stage are completely not medically significant, except in the correct positives are very important. So yeah, I think that what it takes, it's going to maybe take different things in different countries, but as soon as one country or one community, other than the Doria Sharim community, which has already proven that it works, but some other communities, then I think then there'll be a kind of a worldwide competition to save trillions of dollars a year.
All right, thank you very much.
Hi there, Ines O'Donovan, Genesimal Magazine and futurist. As a futurist, I'm very curious to hear your thoughts about how the longevity field will look like in 2050.
2050. Well, one thing that I understand is going to happen in 2050 is going to be the first year in history, or since the Black Plague Middle Ages, that the human population is going to drop rather than go up. And it's going to be extremely top heavy with lots of older people. So we will have a crisis both of fecundity and age. So it will be good timing if all this amazing technology that we have kicks in. I think what we're aiming for is youthful, healthy, vigorous bodies and personalities, so that we will be innovative as we've ever been. And some people are innovative at old age, even without the technologies we're talking about. But anyway, that's what we're looking for, which means that there'll probably be no retirement, certainly no required retirement, and probably no voluntary retirement, because who would want to retire when the jobs are interesting and you've got a whole life experience? You're not in when you started in your career, you were inexperienced and it was hard to get a job. But now everybody's could be experienced as they have more and more time to establish it. I think I think it will be increasingly difficult to tell what age somebody is. I mean, it's already fairly difficult thanks to cosmetics and cosmetic surgery and so forth, but it will become harder and harder.
Like the movie In Time, where he presents his mother, his wife, and his daughter, and they're all the same 25-year-old.
Right. Yes.
Pretty exciting. And what do you think are the most interesting approaches, technologies, except gene therapy, that will bring us to the vision that you've just painted?
Well, I think I like gene therapy for all the reasons I've outlined, but there will be many different technologies. There'll be mechanical. There'll be AI, not just in developing new therapies, but also itself being a part of the therapy. But I think that the key thing is it will be multiplicity. So in pharmacy, it's called polypharmacy, where you have multiple things at once, because I think you have multiple systems that are failing, and it's like whack-a-mole. If you completely got rid of cancer, which is one of the major, 13% of us die from that. That would buy us 2 years. One study indicated a 2-year. If you completely got rid of diabetes, it'd be another small factor like that. So but if you get at the core elements, which still might involve multiple genes, then you might get them all at once. And so I think we should keep our eyes open to, very complex solutions that involve multiple genes, multiple drugs, but they're not any more complex than a jet engine or a rocket or a cell phone. We will do it soon.
Thank you.
Mindful of time, we have only a minute or two left. If you have a few extra minutes, we can extend
Yeah,
that.
sure. I have time if you have time.
Yes.
Perfect. Thank you so much for the great talk. So I wanted to ask you a question. What is your general perspective on gene selection to get rid of the inherited helper mutations in human embryos? Like basically talking about the appearance of designer babies. Do you think it is a good basically application of the technology, or we should be more careful with how we're gonna make it accessible to the general public?
Well, there's a few things to unpack there. I mean, I think the number one is making it accessible to the general public, which includes education and dialogue, education in all directions, not just scientists talking down. So accessibility is an important component. It's emphatically not eugenics, in the sense that the problem with eugenics was the governments of the world, including the United States, enforcing sterilization on people who didn't want it. So it's essentially giving medicine without permission. I think this is different in that this is families want to be able to control their own choice about whether they're going to have a very serious illness in the family or not. And I feel that doesn't answer your full question. Why don't you give a follow-up question?
I would say that, like, from your perspective, like as a whole, general selection, should it be accessible to people who like for example, suffer from HER2 or like
Finer,
HER2?
finer
Yes.
baby.
But like, what about people who do not have like such harmful mutations in the genome, but like just want to maybe make the babies much more intelligent or something like that?
Right. So I think that I mean these are difficult ethical questions, but they're not impossible. I mean, we shouldn't oversimplify them, but we shouldn't say that they're we can't do anything about them. I think that if we don't like, you know, cosmetic therapies, then we should pass laws that prevent cosmetics. You know, why should we have cosmetics at all? If you know, if you're creating a culture where beauty and height and age relate to your employability, or you know, getting spouses and things like that, then we should recognize that that actually is, in a certain sense, life and death. It's not frivolous. Okay. The important thing is to make it safe and effective, rather than having, having fake or inaccurate representations of it. I think we also need to prioritize. It's not quite a zero-sum game where if you're working on X, you're not working on Y, but I think we need to think of, you know, cosmetic and non health related things as slightly secondary to the health related breakthroughs, which we're definitely going to read. But some of them could be accidental side effects of the health gains. If we do get aging reversal of serious diseases, we will probably get aging reversal of a lot of cosmetic features as well. Although it doesn't have to be our top goal, it could come along for the ride. But I think the main thing is we don't want to create a stigmatization of people who are not using medicines, or particular medicines. That they should be, those cultures and individuals should be embraced rather than stigmatized.
That's true. Thank you so much. I appreciate it.
Hello, I am Anton Kulaga, Chief Developer of Longevity Genie and Just DNA-seq open source projects. And continuing the previous question about genetic enhancements, several years ago, your lab published this collections of genes and gene variants that unofficially called Superhuman Genes Database. Actually, we managed to develop an open source module to annotate these variants genomes of people. And we discovered that it requires refinement because some of those superhuman variants are pretty much in most of the genomes. And I'm curious if you plan to continue collecting new data and refining this superhuman database in the nearest future?
Well, I'd be delighted if you sent me a link to that at my email or put it in the chat or whatever, because I'm not, but I'm fairly certain these are not common variants in the population, the ones that are on the web page that we maintain. They are generally rare. But they do exist in the population by definition. Some of them are synthetic that were tested in animals, or and we did publish this in a peer-reviewed paper for in the topic of enhancements for space travel. So we did this together with Chris Mason's group. Yes, we're going to continue to look into that. And I should mention that enhancements, it's not really so much superhuman, it's superhuman relative to our ancestors. We want it to be available to everybody. So our ancestors, for example, lived in mortal fear of about 20 different very serious infectious diseases. And now, thanks to vaccines, we don't even think about them anymore, like smallpox and polio and so forth, measles. And so I think that's an example of enhancement. And that's what we're trying to collect, not just natural examples where you have a rare person in the population. Sometimes there's a reason why they're rare. You know, they have some negative consequence. So they probably have to be implemented in a very careful way, just like everything we've talked about today. But there's also synthetic things that we discover in the process of making animal models, where we can, if we can manipulate by gene therapy, let's say either germline or somatic, or usually both, we can make those animals enhanced with respect to a particular trait or two or many.
Yeah, it's great that looks like you want to continue. Actually, I wanted to also ask one more small question. I'm curious, as today we talked about AI, how exactly is AI used in your lab, and where do you see the bottlenecks? What are the major problems that your researchers face with it?
We use it on almost everything, you know, from ranging from small molecule drugs to, you know, diagnosis to gene design, gene therapy design, genome design for mostly for bacteria so far, protein design. Bottlenecks, I think, you know, some people can't get enough GPUs. I don't think that's our problem exactly, because what we have that other applications of AI don't have is this ability to test millions or billions of things that are pre-filtered by machine learning, but then are post-filtered with something that's 100% accurate, which is the exact object itself. It's not a silicon simulation. So what other bottlenecks we have for AI? I think it's all limited by creativity. That's the same thing. Biotech and computing have gone through exponential, and those exponentials are due to innovation. And I think that, and the AI does not yet provide innovation at the level that humans do, at the level of Einstein and Marie Curie and all the people behind the exponential changes in chips and biotech.
Thank you.
Final question.
Hello, Katya Antonova, architect. My question would be about your ethical consideration arising for genetic specialists using advanced techniques that can influence human traits and future generations. I mean, mostly your own concerns and your thoughts.
Right. So I am generally concerned about everything, especially new technologies. And so since our main passion in business is new technologies, we accompany almost every one of them with a paper on safety and security and ethics. And that's about 36 papers so far from my group. And almost every of these technologies, we precede it with the paper about safety, security, and ethics. Sometimes 2 or 3 papers before we get to the paper that is enabling. So for example, for next gen sequencing, before we publish that, we're publishing ways that you could do the personal genome project that I've already mentioned. Before we do next gen synthesis, 10,000 times more cost effective, we put out a way to do surveillance of orders computationally. Before we did gene drives, we published 3 ways of keeping them contained. And in every case, you should ask, how do we make this reversible? How do we monitor it? How do we, you know, not just self-police, but get the governments interested in policing these things? We have another one that will be coming out in a couple of months. But I think that everybody that's working on cutting edge technology should have a practice of thinking of unintended consequences and publishing those problems and possible solutions first.
I see. Thank you. And regarding changing of human genome and existence in new society when you are basically the same person, I don't know if that's my thoughts correct.
Yeah, as a specific example, changing ourselves, you know, we're constantly changing ourselves. I mean, you know, when we say, you know, we're doing enhancement of intelligence, well, we're doing certainly enhancement via education. I mean, if you look at our ancestors, they didn't have anything like the education that we're getting, and it's almost a lifetime experience for us. I think that you know, the things that we can do with gene therapy in adults is probably very close to as impressive as what you can do in germline. We have a lot of experience with germline engineering of pigs and other animals, and a lot of that can be done in adults. And the thing about adults, the thing about germline is 8 billion of us have already missed the boat for germline. I mean, we're past that, and so I think there's a much bigger market and a lot of enthusiasm for doing it for adult gene therapy, changing ourselves as adults. Yeah.
Thank you so much.
Okay, last question.
Hi, George. I'm Vladimir, co-founder of Unlimited Bio, a gene therapy company from Prospera. So it's been 5 years since you published your paper on AAV combinations in mice. So I was wondering what's stopping it from being brought to primates, as you mentioned, or to humans? Like, when will it happen?
So you're referring to AAV combination therapy for age-related diseases. Is that what you're referring to?
Yeah.
Because we've done a lot of other gene therapies in other applications. So, but this is our main one, is we're really focused on aging because of the cost-effective argument that I said. So that the status of that at Rejuvenate Bio now has 4 gene therapy combinations, gene therapies or combinations in the pipeline for clinical trials for both veterinary dogs and humans, most of them for humans. Yeah.
So when will it happen?
Oh, when will I think the gene therapies should be starting any day now, within the next year or two, depending. The 4 of them are at different stages, so it's happening now, and they probably won't go as fast as the COVID-19 clinical trials went, but I think they're going to go pretty fast. You know, so and certainly the people that are in the clinical trials will benefit immediately. So if it works, which it has so far in the preclinical animal trials.
Yeah, thank you. Good luck with your research. Thank you very much.
Right. Thank you very much, George. It's a fascinating conversation. Could talk to you for hours. I hope you can join us in person next time.
Okay. Yeah.
Appreciate you so much.
Yes. Same. Keep up the good work.
Thank you.